Hello, everybody. My name is Gloria Isabel Giraldo Calderon, and I'm going to give you a brief overview about VectorBase. First, I'm going to tell you what ViewPath is, which is actually the house where VectorBase lives. And I'm going to tell you a little bit about all the tools and data that we have. But most importantly, and where I will spend most of my time, so I will tell you what can you do with VectorBase, which kind of analysis and data mining you can do. I'll also tell you about the ViewPath related sites, and I'm going to share with you where can you get help and documentation to use the website. What is ViewPath? So ViewPath is the home of human vectors, parasites, and fungi. These three types of organisms. And for these three types of organisms, we have both omics and field collected population data. So if you keep that in mind, that ViewPath is the home of these organisms, so this home has the same layout for the different component sites. And VectorBase is just one of those 11 component sites. So if you learn how to use VectorBase, or the core central ViewPath site, you already know how to use all of them. Currently, VectorBase has omics and field collected data, but the way it actually started is that only having genomes. And you know, genomes are both the genes and the regulatory sequences. How are these genomes made? That this is all the genomic sequence. So that genomic sequence gets cut in pieces, and those pieces are called reads. In order to use the genome sequence, we need to put back together those pieces. So it's like a puzzle. But keep in mind that that puzzle sometimes has missing pieces, or we put pieces in the wrong place. So when you come to VectorBase or any other place when you find a genome, the information is not perfect. So if you find something different in the lab with your experiments, you can be right. So we have that sequence of DNA and we put it together, and now we have our puzzle. Now we have our genome assembly. So what is the next step? This genome assembly needs to have the genes. And here I have gene 1, 2, and 3. Depending on the species, we are going to have a different number of genes. But the overview of the steps is the reads, the little pieces of DNA, I put them together to create contigs. The contigs, I put them together to create super contigs or scaffolds. And this step I graded out because it needs to be done in the lab with real chromosomes put together where those super contigs are located in the chromosome. So sometimes the genome projects skip that part. And next, we need to do the gene automatic annotation. As I just said, is locate where each gene begins and finishes. And later, after we do the automatic annotation, some people go and manually check some of the genes. For example, if you work with insecticide resistance and you are interested in genes or mutations that are related to pyrethroids or to DDT. So those are the kind of genes that somebody will come and check manually. And again, the automatic annotation is not perfect, and it doesn't replace a human to come and check each one of those genes, but the human cannot do them all. So that's why the first pass through it is the automatic annotation. This is the top of the VectorBase screen. You will find in the header the site search box. It works like a Google. You just type to find the things, especially if you are new to the website. This will be a great resource for you because it will tell you where you can find things. We will also do exercises in this workshop to tell you how to use the strategy system. And the strategy system is like online shopping, but free. And you are able to run in silico experiments using genomes and transcriptomes, and you can use experimental data. So you can ask questions about your gene of interest using these in silico experiments, and you can share your experiments with your colleagues or put them in your publications. If you click here in tools, you will find all of these available resources, And in this workshop, we are going to tell you how to use the Genome browser. In addition to all the tools that you have available when using VectorBase, also use the data that has been generated in other labs. Currently, we have 44 genomes for both human vectors and other species. And notice here I have an asterisk or wildcard. So we have genomes for mosquitoes, 2 species of Aedes, 23 of Anopheles, and for Anopheles, you know, this is the biological vector Plasmodium that causes malaria, but not all of them are vectors. Some species that are Anopheles that are not vectors have been included for comparative purposes. We also have Culex, a snail, which is not a vector, but is an intermediate host for a parasite called schistosoma. We had a bedbug, which is not a vector, but is an ectoparasite. Flies. We have Glossina, which, as you know, are the vectors of parasites for both human and veterinary importance. And for comparative purposes, Glossina was sequenced with both 2 other flies, Musca domestica and Stomoxys calcitrans. We also have ticks and mites. We also have sandflies, Luxomia and Phlebotomus, which are the biological vectors of Leishmania. We have the body louse, Pediculus, and we have the kissing bug, Rhodnius. Where can you get a summary of all these genomes and the version in which they are, the strain, and other details? You go to the website, click on Data, Organisms, Genome Information, and Statistics. In addition to the genome, we also have 15 other different types of data. Where can you find them? Go to Data, Datasets in VectorBase. One of these 15 types of datasets are transcriptomics datasets, and we have microarrays and RNA-seq. And because microarrays used to be the norm before, we have a few of them, but most recently we've only been adding RNA-seq datasets for all of these species, but mostly we have for Anopheles and Aedes. However, if you're interested in any other species that is within the scope of VectorBase, vectors related with human disease or species of interest for comparative purposes, let us know and we'll be more than happy to add more datasets. So what is the difference between having these datasets here and in NCBI or MBLI? We have all this data that has been generated in a lab and we run it under the same pipeline, and that's how you can use the strategy system, because you are interrogating real data generated under many different experimental conditions. These are all the other 15 types of data sets. So in addition to the genomes, compounds, enzymes, Interpro, which we use for the demo for the strategy system, PubMed, PubSet records, gene, non-redundant proteins database, immune epitopes, metabolic pathways, ontologies, orthology and synteny, protein database, taxonomy, and this is the one I show you, DNA, microarrays, and RNA-seq. And with this, I'm going to end. So if you find VectorBase useful for your research, we will really appreciate if you will make this a resource that is free to you, but our founders need to know that you are using it. So it could keep being not only free, but available. So for example, if you use the strategy system for your analysis, make the analysis public. Share them with your colleagues, not only with your research group, but with everybody else in your poster, your publication, your talk, or your thesis. Use the screenshots from the website, anything that you like, not only the strategy system, but anything. Use our logos. Where can you find them? In the About menu, what is VectorBase? And please reference the resource. So if you go to About, Publications of ViewPath sites, the most recent publication for VectorBase is from 2015. So we'll really appreciate if you can do this. So more people get to know about VectorBase and use it, or we know and our founders know from our users how they are using VectorBase. Once this workshop is over, we want to help you continue using vector-based resources. So you can come here to contact us and send us an email, or you can click here in help, learn how to use ViewPath, and you can request virtual events. You can watch our webinars, which are all available on YouTube. This is a workshop, so this is what you are doing right now. More learning materials in addition to what you are doing in this workshop. And here you will find all the methods, all the pipelines that we have used to produce the data sets that you have used. Other related sites. So in addition to VectorBase, you can also use host database, where, for example, if you're working with, let's say, Plasmodium, so some monkeys infected with malaria, and you want to know different stages of the disease, what are the genes that are turned on and off, those kind of data, you will find them here in HostDB. OrthoMCL is the pipeline that we use to identify all the homolog genes, orthologs and paralogs for all the genomes. Microbiome database, the name says it all, it's about microbiome data, and we're starting to have data from arthropods. Viper and Patrick are essentially as all the other 11 sites from ViewPath are genomic centric, but Viper is for viruses and Patrick is for bacteria. This is all the team. So in ViewPath, we are about 65 people from different institutions in the US and the UK. So please send us an email to help@viewpath.org or click and contact us. I would love for you to try the exercises, and please join us for the live sessions or send us an email if something was not clear or you need help with the exercises. Hope to see you later.